Hi everyone, so today's lecture is called Inborn Errors of Metabolism. So in the previous lecture, we saw how metabolic reactions can be chained into metabolic pathways, and metabolic pathways can be integrated by the body so that the body is able to respond to every single situation that the body is going to face. So with today's lecture, we're going to evaluate what happens if there are problems with the enzymes that are responsible for the enzymatic reactions within the metabolic pathways. So we're going to talk about diseases called inborn errors of metabolism. So we're going to describe these disorders, and then we're going to look at a few examples, and we're also going to discuss how these disorders can be treated or prevented. So first of all, inborn errors of metabolism can also be called heritable metabolic disorders. They're heritable because they're passed down through generations, and they're metabolic disorders because what's affected is actually the metabolism. So there's a problem with one of the enzymes in a metabolic pathway. So therefore, these are called heritable metabolic disorders. So we know that in the DNA, we have the storage of all the information required to produce proteins, and we know that enzymes are proteins. So what happens if we have a mutation, an alteration in the sequence of the DNA? Well, that mutation will translate into an alteration of a protein. And the most frequent changes in the proteins caused by mutations in the DNA are amino acid substitutions, truncation of the protein, or changes in the expression level. And from a functional perspective, all these changes will cause a change in the protein activity and or a change in the protein regulation. So since the problem is at the DNA level, so it's a mutation, this problem is transmissible. So therefore they are heritable disorders. And since the protein which is impacted by the mutation is an enzyme, they are metabolic disorders because the activity or the regulation which is altered is within a metabolic pathway. So therefore, again, this is why they're called heritable metabolic disorders. So before moving on with the disorders, it's important to look at some concepts of genetics. So the first one is karyotype. And karyotype is the number and appearance of the complete set of chromosomes within a cell. And the karyotype presented here is a human karyotype coming from a human cell. And in this case, we have a male. So the first thing you'll notice is that we have 22 pairs of chromosomes. And I'm talking about pairs, but because for each chromosome, we actually have two copies. One chromosome coming from mum and one chromosome coming from dad. So this is very important because this means we have two copies of each single gene. For example, if we have one gene on chromosome 2, we will have two copies of that gene. One copy is on chromosome 2 coming from mum, and another copy is on the chromosome 2 coming from dad. So this is true for the 22 pairs of chromosomes. And then we have an extra pair of chromosomes, which is the sex pair of chromosomes, which define the sex of the individual. So in the case of females, we have a double X, whereas in the case of the males, there is an X and a Y. And you can see here that the situation is a little bit different because the female has a perfect pair of chromosomes. So there are two X's. So in females, all the genes that are on the X chromosome are in double copy. Females will have two copies because one will be on the X which comes from mum and one will be on the X that comes from dad. But as you can imagine, this is not the case with the males because in the case of the male, we don't have the perfect pair. We have an X and a Y chromosome. So the two chromosomes in the pair are different. So in this case, there are genes that we only have in one copy because the chromosomes are different. So they carry different genes. So another important concept from genetics is that of dominant and recessive mutation. So the dominant mode of inheritance means that we only need to have one copy of the mutation to get the phenotype, while in the case of a recessive mutation, we need two copies of the same mutation to get the phenotype. So let's look at the example of dominant mutation, seeing that we can see this at the top of the slide. So the mutation is depicted in red, while the sequence that is not mutated is in yellow. And this sequence that is not mutated is called the wild type sequence. So you can see that the healthy individual on the left-hand side will have this healthy sequence, the wild type sequence, on both the two chromosomes, the one from mum and the one from dad. The individual in the middle, however, will have the wild type sequence from dad, and the mutation will be inherited from mum. And since this is a dominant mode of inheritance, the mutation is dominant, so therefore this individual will be affected with the disease. So we will see the phenotype induced by the mutation. Then, if we look at the third individual on the right-hand side, this individual has two mutations. So the same mutation that is present on the chromosome from dad is present on the chromosome that comes from mum. So there are two copies of the mutation, and of course this individual will be affected. Then if we look at the bottom situation, the recessive mode of inheritance, so the bottom of the slide, the situation is different. So again, we have a healthy individual on the left with both the two sequences which are wild type. Then we have the individual in the middle, in which the sequence that comes from dad is wild type and the sequence that comes from mum is mutated. So the mutation is only coming from mum. There is only one copy of the mutation and this individual is healthy. They do not present the phenotype, so they do not suffer from the disease because the mode of inheritance is recessive. However, these individuals are carriers of the mutation. Okay, so they don't suffer from the disease, but they are carriers of the mutation. They have the mutation, but they do not display the phenotype. Then we have the third individual on the right-hand side that has two copies of the mutation. So this individual has inherited the mutation from dad and also from mum. And since there are two copies, this is a recessive mode of inheritance and the individual is affected. So we do see the phenotype here. So going back to inborn errors of metabolism or heritable metabolic disorders, I just wanted to draw your attention to this review dated 1963. So they're describing inborn errors of metabolism, and they're actually talking about the first time the term was used, which was actually back in 1909. And at that point, only 4 disorders were known. Then in this review from 1963, they're describing 50 inborn errors of metabolism. Whereas nowadays, in present day, we know that there are way more than 50, but luckily the majority of these conditions are rare. But if you go back to the first description of inborn errors of metabolism in 1909, you can see that the way they're describing the disorders is actually correct. They say that the disorders were present from birth, lasted through life without worsening or improvement, and said that they were present in other members of the family. So the reason they saw these present in other members of the family were because the disorders are heritable, and the reason they're present from birth and last through life is because they're due to a genetic mutation, due to an alteration in DNA. So classical inborn errors of metabolism have a mechanism which is having a mutation in an enzyme in a metabolic pathway, and therefore as a consequence of the mutation the enzyme is not working properly anymore, and this is shown in box 1, molecular mechanism 1. However, there are other metabolic alterations in which we do not have alteration of the enzyme, but rather an alteration in the regulation of the pathway. So the metabolic pathway is not integrated correctly anymore in the metabolism, and that's mechanism 2. So let's see what happens. In the first scenario, we have an alteration in the activity of the enzyme. So the enzyme is not functional anymore. Here we have a pathway, so we go from compound 1 to compound 2, and then to compound 3. And we have the enzyme which is able to catalyze the conversion of compound 2 to compound 3. And we call this enzyme the wild type enzyme because the sequence is correct. So there's no mutation. The enzyme is working fine. Then we add the mutation. And so from the wild type enzyme, we go to the mutated enzyme. And you see the compound 1 is transformed into compound 2. But then since there is a mutation in the enzyme and the enzyme isn't active anymore, then compound 2 cannot be converted into compound 3. And now you can start to see why we can have a disease in this situation, because we accumulate compound 2 and maybe compound 2 is toxic. And the other possibility is that we need compound 3, so we need compound 3 for the body to function, but actually since the enzyme's mutated, we can't produce enough compound 3, and compound 3 is really needed, so we get a disease. So these are the inborn errors of metabolism of this first mechanistic group that we're going to look at in this lecture. So these are examples of the classic inborn errors of metabolism where the mutant enzyme is not functional. So then we have the second group of disorders where the problem is not in the function of the enzyme, but in the regulation of the metabolic pathway. So the pathway is not regulated. So it's not correctly integrated into the metabolism of the body. So for example, if you look at the left-hand side of the slide, in this situation, there's no inhibition over the enzymes in the pathway. So the pathway is always on. And this means we have an accumulation, for example, of compound 3, and compound 3 could be toxic. On the other hand, if we look at the right-hand side of our slide, we might have a block in the activation of the pathway. So the enzyme that is regulated within the path is not switched on correctly. So the pathway is not on or is not on enough. So then we've got the production maybe of intermediates, an accumulation of intermediates such as compound 2, but maybe we don't have production of enough compound 3. And you can see that all these situations could potentially generate a condition or disorder. And we're only going to talk about one example of these type of disorders where there's alteration of pathway regulation. An example we're going to talk about is familial hypercholesterolemia. So we'll talk about that later on. So if we go back to our first group of disorders, the classical inborn errors of metabolism, we'll look at the first disease, which is albinism. So you can see here that we have phenylalanine, which is an amino acid, and this can be transformed into tyrosine, and also we eat tyrosine with the diet. Tyrosine is another amino acid and can be used for different things. So for protein synthesis, for thyroid hormone synthesis, and tyrosine can also be transformed into DOPA. And DOPA is a really important molecule because DOPA can be transformed into dopamine, which is a brain neurotransmitter, but also into melanin, which is a pigment. So if the transformation of DOPA to melanin is impaired, then we have albinism. So albinism is a rare condition that affects around 1 in 20,000 people worldwide. And as you can see from the right-hand side of the slide, it doesn't just affect humans, it affects animals as well. The characteristics are the lack of pigments, so the fur, skin, hair will be white or extremely pale, and there are also problems with the eyes. And you can see here photographs of Johnny and Edgar Winter, and these are famous rockers from the 70s that were affected by this condition. So as we said, the problem here is that there's poor or no production of melanin. So there are degrees of albinism. So some people have no production of melanin at all, and some people just have a reduction in the production of melanin. It depends, there's a sort of spectrum. The correct name is oculocutaneous albinism because the most visible problem is of course the lack of pigment in the skin, hair. But there's another problem, which is with the retina, because the melanin is also important for the correct development of the retina. So there are 4 genes that can be mutated, and therefore 4 enzymes that can be altered in their function. And these alterations are able to cause albinism. And depending on which mutation, which gene and which enzyme is affected, we have 4 different types of albinism. So briefly, tyrosine is transformed into DOPA, and then there's another transformation of DOPA to dopaquinone. And this is the reaction that is impaired if there's a mutation in the enzyme tyrosinase related protein 1 or tyrosinase. There are 2 other enzymes that can be mutated in albinism, SLC45A2 or protein P. And we know that the mutations of these 2 are related to albinism, but we don't know much about these proteins in terms of what they do in the synthesis of melanin. So there's research going on for those 2. So albinism is a disorder, it's a condition, but it's not actually life-threatening. So now we're going to explore another condition which is life-threatening if it's not controlled. And this condition is called phenylketonuria. And again, this is the same diagram you've seen before. So phenylalanine is transformed into tyrosine, and we see that if tyrosine is converted to DOPA, and then DOPA is not converted to melanin, then we have albinism. But what happens if phenylalanine is not transformed into tyrosine? Well, in this situation, we have phenylketonuria. And the problem here is that phenylalanine is an amino acid we need to produce proteins. However, if we have too much phenylalanine, we need to degrade it, like everything. So the body has to be able to degrade the molecules that aren't needed. And the degradation, the breakdown of phenylalanine goes through tyrosine. So phenylalanine is transformed into tyrosine, and then tyrosine is liberated. But if we don't have the enzyme which is catalyzing the transformation of phenylalanine to tyrosine, we then accumulate phenylalanine and we cannot eliminate it, and that's the problem. So basically this slide shows the path where phenylalanine is converted to tyrosine, and you can see there's a hydroxylation on the ring. But if there's no active enzyme mediating this hydroxylation, then we have the disease. And the enzyme is called phenylalanine hydroxylase, and the disease is called phenylketonuria, which is also known as PKU. So phenylketonuria, or PKU, is a rare condition, and we have seen it's caused by the mutation in the enzyme phenylalanine hydroxylase, which leads to the accumulation of phenylalanine. And accumulation of phenylalanine is toxic to the brain. So you can see the problem here. So people with PKU introduce phenylalanine in with their diet because phenylalanine is an amino acid that we find in food, and they're not able to eliminate it. So some of it will be used, but the majority of it will be accumulated with no possibility of being broken down. So this accumulation, little by little, is toxic for the brain. And the situation here is very peculiar. So babies with PKU, they're fine at birth, and the problem kicks in within the first few months after birth because the baby, if they're not recognised to be affected by PKU, they'll be fed on normal milk. And then they'll start to take in phenylalanine with their diet. And they won't be able to degrade the phenylalanine, and so phenylalanine will be accumulated, and this will cause damage to their brain, and this kind of damage is irreversible. So people with PKU that aren't diagnosed at birth, they will go on to develop intellectual disability. This, of course, is the extreme scenario, and it's an irreversible situation. However, if we manage to diagnose PKU at birth, then there's the opportunity to treat babies with PKU and prevent the intellectual disability. So in fact, if a baby is diagnosed immediately at birth with PKU, instead of feeding them with normal milk, we can change the way we feed them. So we can start treating them so they will not accumulate phenylalanine and allow their brain to develop normally. So if we spot PKU immediately at birth, we can treat the baby and the person will go on developing totally normally and will live a normal life. However, if we don't detect PKU at birth, then there's a problem because the baby will go on to develop intellectual disability and this brain damage is irreversible. So the change in diet starts at birth, as we said, and this will go on through the entire life of these people. They cannot eat phenylalanine because they can't break it down, they can't metabolize it. And so their diet is pretty much a vegan diet. So you can see they're allowed to eat the foods that are close to the center of the diagram. So that's vegetables and really low protein foods. They're not allowed to eat what's around the circle. So they can't eat meat, fish, they can't eat eggs, they can't drink milk. So you can see it's really like a vegan lifestyle. So I told you it's very important that PKU is recognised as soon as possible at birth so that we can immediately switch the diet to the babies and preserve their brain function and let them develop normally. So it's very important to test these babies at birth. And PKU is not the only disease that is important to spot immediately. There are other metabolic conditions. So in the UK and actually in other countries, for example in Europe, there's a service that's offered to all the babies that are born. They are screened for 6 inherited metabolic diseases, as well as including PKU. And you can see that the nurse is taking a drop of blood on the photograph from the baby, and the drops of blood are spotted onto a card. And these drops will be analysed to detect whether these babies are okay or if they have metabolic conditions so that we know about them and the babies can be treated immediately. So the screening for PKU was originally done with a test called the Guthrie test, which isn't performed anymore, but is interesting. So in the Guthrie test, we need to have a strain of E. coli which is phenylalanine negative, meaning that the strain of E. coli is not able to produce phenylalanine. And we need to supplement the phenylalanine through the growing medium. So we plate the bacteria on an agar plate, and then we supply the bacteria with phenylalanine in the growing medium, and the bacteria will grow and cover the entire dish. Then we take another dish in which we have minimal medium without phenylalanine. Then we take a little bit of bacteria and spread them on top of the second plate, which of course is a minimal medium, so there's no phenylalanine. So the bacteria can't grow, but they persist. So the bacteria will still be there, but they don't grow, they don't duplicate. And then we take the blood spots and put them on top of the plate. And you see that if in the blood there's phenylalanine, then the bacteria will start to absorb the phenylalanine, and this will promote their growth. So then if there's a colony growing around the blood spot, we know that in the blood drop there's phenylalanine. And if there's phenylalanine in the blood, it's because the person is affected by PKU. Okay, that's why there'd be phenylalanine in their blood. Okay. So as we said, nowadays the Guthrie test is not performed anymore because it's expensive and time consuming. So what's used instead is a high throughput technique, for example, tandem mass spectrometry. But I'm not going to go into the details of this technique, but just briefly, the technique allows you to check a biological sample and check the composition of the sample. So you're not just detecting one single component at a time. So in the Guthrie test, you'd only be able to spot the presence of phenylalanine. But with this analysis, with this machine, you can check multiple molecules at the same time in the same analysis. So you prepare your sample, you inject the prepared sample into the machine, and then you'll get a report out at the run of your sample, reporting different molecules, among which is phenylalanine.